My name is Qiangrong Liang. I came from New York Institute of Technology, College of Osteopathic Medicine. My research has been focused on diabetic heart disease. As we all know, diabetic patients have increased heart failure. They are more susceptible to heart disease. My lab is asking a question, why these people are more sensitive to heart disease. One theory for the increased possibility for heart disease in diabetic patients is the oxidative stress. It's increased ROS, reactive oxygen species production. That's the major theory. Actually, this theory has been supported by overwhelming evidence from animal studies and in human studies. But the trouble is several clinical trials using antioxidants to treat heart disease patients. So they didn't observe the same result as in animals. That challenging this free radical theory for increased heart disease in diabetic patients. So my thinking is the oxidative stress, so ROS generation, so that could be still the true mechanism. However, the antioxidant they used can only scavenge or antagonize existing ROS, the reactive oxygen species, so-called ROS. But they didn't really stop the continuous generation of ROS, which is mainly from mitochondria. So mitochondria actually has been observed to be a really important component in the disease process of heart disease in the diabetic patients. Mitochondria are easily to be damaged. So when mitochondria is damaged, they produce more free radical. So that will cause more damage. So that's why the antioxidant could not work. That's because you didn't take care of the injured mitochondria. So so my focus is on how to remove those injured mitochondria to stop the continuous production of free radical generation. So the pathway is actually called autophagy. So that's the pathway responsible for the degradation of mitochondria. So we hypothesize if we enhance autophagy in the heart, it could reduce diabetic damage. We did this in animal studies, but surprisingly, we found that we increase autophagy in the heart, decrease autophagy in the heart, we just get opposite result. So indicating autophagy actually is not protective. Instead, they are detrimental to the heart in type 1 diabetes. So what is the reason? So the overwhelming actually assumption is that autophagy actually the equal autophagy, including myself in the past, I equal autophagy as mitophagy. That is the process to degrade mitochondria. Actually, the autophagy and mitophagy may not change in the same direction. So when you manipulate autophagy, actually you may not induce required, desired change in mitophagy. So that's my actually a new hypothesis. Actually, we did this so in cell culture, in animals. So this really suggests. So when you manipulate autophagy, you may not see the desired result. So now we target at specifically mitophagy. We made a mitophagy specific reporter. So it's really very convenient, very easy to use. It can report the occurrence of mitophagy in cell and in the heart. So that's my talk will be focused on today. So hopefully, so this new tool will add to the current technology to help scientists to understand better the role of mitophagy in the heart. So hopefully, so in the next few years, I think there will be an explosion of literature indicating it is mitophagy, not autophagy, which is more important in the pathogenesis of heart disease in diabetic patients.